Hi, it's Dr. Barr, and I love me some histology. We're going to talk about neurons. This lecture, we're going to correlate with some anatomy lectures related to the nervous system. Anatomy has already introduced some of these concepts to you. Histology will further reinforce these anatomic concepts and emphasize more of the microscopic morphology along with some cell biology. Recall and utilize the concepts from the Intro to Histology lecture as we move forward. This lecture, we're going to visualize and re-emphasize the structures via light and electron microscopy. From anatomy lectures, you know by now the organization of the CNS and the PNS, and also the different types of neurons. The neuronal classification, this is based upon the number of processes. So the multipolar, which is commonly tested and commonly seen on histology, going to have one axon and more than two dendrites. These are usually seen with motor neurons, either with somatic or visceral organs, as well as interneurons, and their role is basically for impulse conduction using their axons. The bipolar neurons have one axon and one dendrite. They're very specialized and occur more commonly in special senses like taste, smell, hearing, sight, and equilibrium. Pseudo-unipolar are commonly also tested. These axons divide close to the cell body into two branches. So it's basically one cell body in the middle, and then you have your axons on either side. This is usually seen with sensory, dorsal root ganglia, and cranial nerve ganglia. First, an example of the typical neuron, which has some key functional components. And the classic example we're going to use is a motor neuron, which is a multipolar cell. It has the cell body, which is the most prominent part of the cell, containing lots of organelles. You can also recognize the nucleus, which can be located within that cell body. The dendrites receive information. They can, in this case, it's multiple dendrites. And then you have axons, which is only one, transmitting the information. Before the transition from the cell body to the axon, you have this axon hillock, which is a clear region in the cell body, which lacks any cytoplasmic organelles, and it's a key distinguishing landmark to identify the difference between axons and dendrites. Figures and diagrams already. Now let's focus on the histology. Here are some examples of light microscopy images at various magnifications. This is pretty much a high power magnification of a multipolar neuron, one axon, multiple dendrites. You can see the multiple dendrites sticking out there. The images show microtubules, which are present more in the upper image there as well as in the lower image. What you're seeing are these very fine lines. Can you see the fine lines like streaks within the cell body extending into the axons and dendrites? So I want you to distinguish between microtubules versus neurofilaments. Neurofilaments are intermediate filaments, which are key cytoskeletal proteins. They basically give the cell its shape. Right? Microtubules are more commonly seen with movement of things, but both structures can be involved in exoplasmic flow. Both structures, like I mentioned before, can be present in the cell body, axons, and dendrites. These neurons in general are present throughout the CNS. Light microscopy, I want you to be able to identify the nucleus with the prominent nucleolus, that's dot in the center of the nucleus. Of course, the cell body is the predominant portion of the neurons, containing Golgi bodies and rough endoplasmic reticulum. Now, keep in mind, from the Intro to Histology lecture, Golgi body and RER are not really recognized visually, right? However, some cells, because these cells are protein producing, they're going to have a lot more abundant RER. And the special stain, which is a toluidine stain, is identifying these Nissl bodies, which reflect the RER and the ribosomes attached to them. So you can see that there's a lot of it, making the cell cytoplasm look very clumpy. So we can't really see the Golgi body per se, but do know from cell biology that in order to make protein, you need the rough endoplasmic reticulum with the ribosomes, right, for translation, and protein production, as well as Golgi body to package these proteins. And talk about the histology of the spinal cord. Keep in mind that when you look at the spinal cord, and particularly with this kind of staining, the lighter area is going to be the white matter, composed of nerve fibers, basically myelinated axons, that pale area. The denser, bluer, purplish area, the center, you can have your dorsal and ventral horns. They're going to contain the actual neuron cell bodies. So don't forget, dorsal is sensory, ventral is motor. The way I think of it is like if you're petting a cat, right? The back of the cat, you're petting the cat, so they're feeling the petting on the back. And then, of course, the ventral area is where they have the legs. And so that's where they can either kick you from too much petting or run away. And then you look at the roots. You don't forget you have your ventral and dorsal roots. Now, the ventral roots have the motor neuron axons, the axons. The dorsal roots actually contain neurons, okay, actually contain the sensory neurons. We're going to go into more detail in subsequent slides here, but do know that slight difference. Motor neuron cells are actually in the horn, right, anterior horn, but they extend their axons into the ventral roots. The dorsal roots can contain sensory neurons, which are your pseudo-unipolar cells. Looking at the dorsal root histology, dorsal root is also called spinal ganglion, dorsal root ganglion. It's a little bit more bulbous, and the reason why it's a little bit more chunkier and round because it contains sensory neuron bodies, those pseudo-unipolar cells that I mentioned earlier. Don't forget, tie this in with anatomy, that what is their role of these cells, right? Sensory neurons, right? Sensory stimulation. So they're receiving pain, touch, vibration, proprioception, and temperature, right? And they're going to mobilize and sense from the PNS and contribute to communicate with the CNS. Now, the pseudo-unipolar fibers, those ones that are going towards the periphery, right? Those ones that are going through the periphery, here is the cell body. And then if the ones that are going towards the periphery are going to leave through the spinal nerve and run alongside with the motor fibers. As you can see here, the motor fibers are the red lines. They head, the ones that are going up towards the spinal cord, right? They head towards the gray matter of the spinal cord, which will then communicate with other cells to go upstream. More pretty pictures, you have a low power magnification of the spinal ganglion, this bulbous round ganglion here that's cut in cross sections. I want you to see the cross section of these cell bodies, which are the sensory neurons. So this whole ganglion here, okay, containing multiple sensory neurons. This is a low magnification view, okay, where you can see other nerve bundles next to it, axonal bundles next to it there, and this is adipose tissue, which is more bubbly there in that appearance. Now also you can have within the ganglion support cells called satellite cells. These are metabolic support cells to the sensory neurons because, again, they need nutrients, they need communication to make sure that they're very healthy. So this is low power magnification on the left-hand side. This is high power magnification view on this side where you're actually seeing the cross-section of these sensory neurons, which are the larger cells with the nucleus in the center. And then all of these other nuclei, smaller nuclei that are surrounding these sensory neuron cells are your satellite cells. Don't forget, satellite means peripheral, right? Peripheral. And so that's what they do. They are metabolically and support cells and located peripheral to the sensory neuron cells. Histology of the ventral horn, you can see again the motor neurons in the ventral horn have what we call polygonal shaped cells, which are kind of like multi-sided. They're multi-sided because remember they have multiple dendrites and only one axon. So here you can see a dendrite, and here you can see the motor neuron. Here are the support cells, also again similar to satellite cells that you saw earlier. So this is a standard H&E image there. On the right hand side, like I showed you earlier, this is a Nissl stain, which shows you the Nissl bodies. And remember again, what is the composition of the Nissl bodies, right? RER and ribosomes. And so here's a dendrite which do not contain Nissl bodies, right? And so you see this region right here, which is the axon hillock. So that's the area that's lacking organelle, even lacking Nissl, right? And then that's going to go into an axon somewhere next to it. We discussed about the cell body and the different types of neuron, mainly the pseudo-unipolar cells and the multipolar cells. Let's talk a little bit about synapses, how these cells are communicating with one another or with target cells. So that's the key thing here. If neurons are going to communicate with each other, like the ganglion sensory neurons communicating with the spinal cord, right, they can either have communications via axodendritic, axosomatic, or axoaxonic. Basically, you're just taking the words, right? Axoaxonic means axon to axon. Axosomic means axon to the actual cell body of the neighboring cell. And then axodendritic is the axon communicating with the dendrite of that cell. The next slide, we're going to talk about the communication or synapses between an axon and an effector or target cell. And the communication between axon and target cell or effector cell comes in two flavors. Electrical synapse does not require neurotransmitters. This can is commonly seen in invertebrates, but in mammals, it can be present in smooth and cardiac muscles. Basically, electrical conduction or axon potentials going down, you have these connector structures, which are the gap junctions. You have an opening in the center, basically, to allow for the action potential to go right across directly. Now, the other synapse is going to be a chemical synapse, and the chemical is neurotransmitters. So you have an action potential coming down the axon. The neurotransmitters are released out of the synaptic vesicles, crossing a cleft and going to your target cell there. The target cell may, again, receive that neurotransmitter, and the effect of the neurotransmitter can be either excitatory or inhibitory. For dive in the chemical synapse, you have a presynaptic knob, which is basically a terminal axon. Within that terminal axon, you have your membrane-bound vesicles containing your neurotransmitters. And so that will, again, cause some type of calcium voltage gauge channel here to contribute to regulate the release of these neurotransmitters, which will cross the synaptic cleft. And then once across the synaptic cleft, they will reach some type of receptor site on the postsynaptic membrane of that target organ, say like the muscle. And then it will cause opening of certain sodium channels to allow for the action potential to trigger the function on that target organ. To more detail once we get into the muscle section of the physiology and histology. However, at this point in time, I do want you to know that part of the nervous system, right, you can see in the top image here, you have your axon coming down as part of the motor junction. You have your neuromuscular junction here where the axon is going to terminate into the presynaptic terminal containing the synaptic vesicles. And so what are we going to see on electron microscopy, right? We have the synaptic, here's the axon terminus here. All these dots in the axon terminus are basically your synaptic vesicles. They're coming towards the membrane surface, which they will be released. And this area right here in between is going to be your cleft, your synaptic cleft. So you can see that on this end here is going to be the postsynaptic muscle cell. So here's the presynaptic axon terminus, the synaptic vesicles, neurotransmitter will cross the synaptic cleft, reaching the surface membrane acetylcholine receptors. Once they are targeting and opening they tie to the receptors, they will open the sodium channels, which will eventually generate into some type of action potential for muscle contraction. So just to recap, we've gone over the normal neuron anatomy, comprised of cell body, axon, axon hillock, and dendrites. Don't forget the Nissl bodies, which reflect the ribosomes, because this is a protein-producing cell. You have a neuronal classification of multipolar, bipolar, and unipolar types. We also discussed the modes of transport from synapses, as well as the different types of electrical versus chemical type of synapses. On that note, email me or contact me if you need any questions answered or clarifications, and I will see you at the next lecture. Thank you. Bye!